Welcome to my talk, which is about the wandering glider, which is a migratory odonate. And what I did is to compare the mitochondrial genomes of migrational and non-migrational populations of this really globally distributed dragonfly. First of all, I'd like to thank a few people, which are namely Christoph Bleidern from the University of Göttingen, who helped me a lot with the bioinformatics, and to thank Rob DeSalle from the American Museum of Natural History, where I could conduct big parts of my research, and Ingrid Avial, who offered me a lot of samples that I was using in this I will use talk. So, first of all, to give you a little introduction into the wandering glider. The wandering glider is the world's most abundant dragonfly, and it's distributed all over the world on all continents except Antarctica, and it has outstanding migratory behavior. It covers every year on a multi-generational route up to 18,000 kilometers, which is outstanding and not reached by any other insect. And does this in order to follow the reins of the intertropical convergence zone, which shifts every year, and it does this in order to put their eggs in the emerging ephemeral waters. And by being such an outstanding migratory insect, even transoceanic and transhimalayan migration is observed. And at the same time, it is the only odonate on the really remote Easter Island, which belongs to Chile and is about 1,000 kilometers away from England. And this population over there doesn't show any migratory behavior, and even morphological differences are observed already. So there's ongoing speciation, and genetic isolation is observed, and of course geographic isolation. So what I was wondering and what my talk is about is three main questions. I had a look at my genomes from migratory and non-migratory populations of Pantala flavescens, so I compared them intraspecifically and even compared them interspecifically with other odonate mitochondrial genomes. As a second aspect, I had a look where there are different selection pressure acting on migratory and non-migratory populations of the wandering glider, and a bit as a shot in the dark, I had a look whether there are endosymbionts involved, because as I told you in the beginning, there's ongoing speciation, and often endosymbionts are involved when it comes to speciation. So starting with my taxon sling, I had 4 from 4 different localities, and 3 of them were from continental and migratory population, which is in Tanzania, which is in China, and from Ecuador. Then and as the 4th one, I have a non-migratory population from Easter Island, which belongs to Chile. So there, from these localities, I have my mitochondrial genomes, and
Felix. Felix.
Yeah.
So, sorry to interrupt. We have some audio problems, and sometimes it helps if you turn off the video. So because I only hear every second word. So let's try that. Maybe it helps if you turn off the video.
Okay. Yes. So is it better now?
Let's see. Let's see. I hope so.
Okay. So hoping it now. You have my mitochondrial genome localities here. This is 4 localities from migratory and non-migratory populations. So the continental ones are migratory, and the last one from Chile, Easter Island, is non-migratory. And as a second aspect, I told you I have endosymbionts. I checked for endosymbionts. I have in total 11 populations from Namibia, in Kenya, Tanzania, and Madagascar, and from Australia, and the other ones are from the populations are already tested for my mitochondrial genome analysis. So just short, do you hear me better now?
It seems like that. It seems I hear you a bit better.
Okay. Okay. So sorry for the technical issues. So coming to my results, I have complete 4 mitochondrial genomes, and as you see here, this is a set of one Tanzania, and all of my 4 mitochondrial genomes have the typical set of 37 genes and a high AT nucleotide content, and they differed in length about 250 base pairs. So we have length polymorphisms comes to mitochondrial genome, but even when it comes to the control region and in the tRNAs, and especially in the protein coding level, which is interesting because the Easter Island population from Untitled has the 4 gene, which is about 200 base pairs shorter than the protein coding genes of NRD4 from the migratory populations. So intraspecifically from migratory and non-migratory populations, we have length polymorphisms within the whole mitochondrial genome, within the control region, and even a truncated NRD4 gene shows the domain. Which shows a missing domain. So the NRD4 gene normally has 2 functional domains, and the NRD4 gene of the Easter Island population has a 1 missing domain. Interspecifically, we have most compact mitochondrial genomes in the shortest control region, even if they differ in length, but all of them were the shortest compared to other odonates. We have many overlapping sequences, but in general, no differences, significant differences when it comes to nucleotide composition. So discussing the first point, we have intraspecific length polymorphisms in odonates, and they are really common in insects, especially when it comes to the control region. And I showed you that there are differences, huge differences in my control regions, from 160 from the Easter Island to about 350 from the Ecuadorian specimen. But these differences are often observed, but really remarkable is that we have differences in the protein coding genes and a missing domain in the NRD4 gene. And the NRD4 gene is, as all of the mitochondrial genes, are part of the oxalose pathway, and the oxalose pathway is responsible for the majority of the energy that is needed for locomotion and therefore needed for long distance migration. So even if there's a functional protein still possible, obviously because these guys fly, it should have an impact on its fitness. So what I did as a second aspect, I had a look at the selection pressure. And what we can see here is that we have my 4 populations from China, Ecuador, and Tanzania, which are all of them migratory. And the 4th one I have from Easter Island, which is non-migratory. And here you can see the 13 protein coding genes. I tested for purifying selection pressure, and every value that is higher than 1 indicates purifying selection pressure, and every red value indicates even significant high selection pressure. And what we can see here is that every 4 of the 4 populations shows in all of the 13 protein coding genes really high purified selection pressure, and all of them more or less show the same values. So we have really high pure selection on all of my genes, and I didn't show you the results yet, but there was even a strong bias in codon usage and a strong AT bias in the codon usage in all of our 4 populations. And this really strong AT bias is common in odonates. We have it reported to be stronger in dragonflies than in damselflies, and it seems to be correlating with its better ability to fly. What is really interesting that we have the really high selection pressure in all of our 4 populations of the migratory and non-migratory ones. And keeping in mind that the selection pressure is the main evolutionary force that shapes the mitochondrial genome, it's really interesting that we still have these polymorphisms, especially from a truncated protein coding gene, that these polymorphisms are able to establish because we have a really high selection pressure. And I think this is only possible through different selection pressure that is acting on the non-migratory population, and together with the result, together with isolation, it could establish. So we have different less selection pressure acting on the Easter Island population that doesn't migrate anymore. And as I told you in the beginning, we even tested for endosymbionts, because endosymbionts can have really high impact or influence. So we tested for different endosymbionts, for different ones, and 2 of them were tested positively, and namely this is Spiroplasma and Wolbachia. So we can see here the 11 population that we tested, and yeah, starting with Spiroplasma, more or less 50% of them, of the populations, were tested positively, and of the specimens were tested positively, but there's no real pattern of infected and non-infected populations. A really different picture we do have when it comes to Wolbachia, because Wolbachia is only present on the Easter Island population, and it's not present in any of the other populations. So this is really remarkable. Yeah, Spiroplasma and Wolbachia are both pretty common endosymbionts in insects and in odonates. And starting with Spiroplasma, as I showed you in the beginning, that Pantella flavescens is globally distributed, and the transmission rate of Spiroplasma is temperature dependent. So I think that these more or less random pattern of Spiroplasma infection correlates with the different temperature profiles that we do have on different in different part of the regions where Pantella flavescens is distributed on. And we do have another picture in Wolbachia, and we can see that it is only present on Easter Island, and I think, or the question is whether this is only evolutionary stable on Easter Island, because we see that there are differences in the mitochondrial genome, and Wolbachia can have a selective sweep on its host mitochondrial genome and lead to genetic isolation, because if populations are infected with Wolbachia and other aren't infected, it could be, or this could lead to that these populations cannot intermate anymore. So we have ongoing speciation on Easter Island, and I think it is really accelerates by its infection with Wolbachia. What we can see here on the bottom, this is a really old picture, already 20 years ago. This is one of the first papers that was conducting research on the Easter Island population of Pantella flavescens, and we can see that there are many morphological differences from the continental ones and to the Easter Island population. You don't need to read everything of this, but there's a lot of morphological differences, such as smaller forewings, and they even have a different flight behavior. So these infection pattern of Wolbachia really should accelerate its speciation and maybe has an impact on its population that does not migrate anymore. So coming to my conclusion, in total, I sequenced 4 complete mitochondrial genomes of migratory and non-migratory populations, and more showed syndrome persistence. But nevertheless, on the other hand, we do have length polymorphisms, which are in parts really common, but not really common is that we do have a truncated PCG, protein coding gene, and we do have high selection pressure on all population, and an exclusive Wolbachia infection pattern that is only present on Easter Island. And together with the differences that we do have in the behavior and the phenotype non-migratory Easter Island population, I think that there's a trade-off between being a migratory insect and the establishment of an isolated population that has ongoing speciation. So in order to conduct further research, I think it's really necessary to study life history traits that we had in our talk before. So I think it's really important to study the larval stages of non-migratory East Island populations and migratory populations of the wandering glider, or to study biotic and abiotic factors on the different localities. And I think it's really important to have an inside look with if you perform transcriptomic analysis. So as a finishing aspect, I think that Pantella flavescens, the wandering glider, is a really promising model organism because we do have this non-migratory and migratory populations. So I think Pantella flavescens is a really promising model organism for long-distance migration at the base of winged insects. Thank you.